It's Michael and good morning everyone. Today we continue our lesson on mutation. Okay, so mutation is a change in the nucleotide sequence of an organism's DNA or in the DNA or RNA of a virus. Now we are focused on organism's DNA. Okay, so the definition for mutation would be best for a change in the nucleotide sequence of an organism's DNA. Mutation can arise in a number of ways. Errors during replication or recombination can lead to nucleotide pair substitutions, insertions, or deletions, as well as the mutations affecting longer stretches of DNA. Let me rephrase that. Okay, during DNA replication or recombination, what happened? Okay, the stretch of DNA. Okay, what happened? The nucleotide, DNA consists of nucleotide bases. These nucleotide bases, okay, it can be substituted, digantikan, or inserted, meaning to say the stretch of nucleotide base on the DNA is being inserted with other nucleotide base pair, or deletion, meaning to say the nucleotide base pair on the DNA is being deleted. Okay, and this can affect either a short stretch of DNA or long stretch of DNA. Mutation is a chemical or physical agent that interacts with DNA and can cause mutation. Mutants are new genetic material produced due to mutation, and mutations can be classified into gene mutation and chromosomal mutation. Today's lesson we only focus on gene mutation. Okay, next lesson then we focus on the chromosomal mutation. The idea is, because I don't want you to confuse between gene mutation and chromosomal mutation. Okay, right. How to change the next slide? All right, gene mutation. Okay, also known as small scale mutation. Okay, what is gene mutation? It is the changes in the sequence of nucleotide bases of a gene, right? Usually, okay, gene mutation is referred as point mutation because it occurs within a single nucleotide pair of a gene. Why a single nucleotide pair? Because DNA is a double helix. Okay, on the other strand you have nucleotide base, which is complement with the other on the other strand, so it's a base pair. So point mutation because it occurs only on a single nucleotide pair of a gene and maps to only one chromosome locus. Okay, now gene mutation occurs during gamete formation. Okay, causes the mutant gene or allele mutant, okay, able to be transferred to the offspring. Now there are two types of gene mutation: single nucleotide pair substitutions, okay, and nucleotide pair insertions or deletions, or also known as frameshift mutation. Okay, now class, there is a difference between point mutation and frameshift mutation. Now, gene mutation usually referred as point mutation, but bear in mind there are two types of gene mutation here. Okay, the point mutation itself and frameshift mutation. Okay, now let's get to point mutation or single nucleotide pair substitution. Okay, now a single nucleotide pair substitution is the replacement of one nucleotide and its partner with another pair of nucleotides. Okay, or you can simply say a single nucleotide pair substitution is the replacement of one nucleotide pair with another pair of nucleotides. And then the single nucleotide pair substitutions or point mutation, okay, it may cause silent mutation. Okay, actually there are three. Okay, now we go to the first one. It may cause silent mutation. which is a change in a nucleotide pair that may transform one codon into another codon that is translated into the same amino acids. Okay, example is we have CCG, as you can see from here in the original DNA strain. Okay, CCG over here we have CCG. Okay, on the DNA template strain mutated to CCA. CCA. What happened? The G, okay, this one nucleotide pair is being replaced with this pair. Okay, the G is replaced with A. Okay, or the base pair GC is replaced with the base pair AT. So the mRNA codon that used to be GGC, okay, it was supposed to be GGC, will become GGU. But glycine would still be inserted at the proper location in the protein. Luckily, both GGC and GGU codes for glycine. Okay, so this is silent mutation. No observable effect on the phenotype because no effect on the amino acid sequence. So as you can see from here, the sequence of the amino acid remains unchanged. Okay, so it has no it has no observable effect on the phenotype of that organism. All right, now what happens if the sequence of the amino acids here change? Okay, let's proceed to the other example of point mutation. Okay, so we have covered silent mutation. Okay, mostly this point mutation cause missense mutation. Okay, missense substitution that change one amino acid to another amino acids. So substitution mutations are usually missense mutation, that is the altered codon still codes for an amino acid and thus makes sense, although not necessarily the right sense, meaning to say the amino acids, okay, that is being coded is not the proper amino acid. Okay, it became unwanted amino acid. Now this missense mutation, it has a range of effects, depends on the location, okay, within the protein and the identity of the new amino acid. Now a famous example of missense mutation is the sickle cell disease. Okay, you refer to this diagram here. Okay, now CTC, okay, on the DNA template strain, what happened? Mutated to CAC. Okay, remember point mutation is the substitution of a base pair. Okay, so what happened here? Okay, the T is being replaced with A, as you can see from here. Right, it's A here. So the mRNA codon that used to be GAG, okay, will become GUG. So the mutant beta globin will have valine instead of glutamic acid. By right, they should have glutamic acid in the sequence of the amino acid in the polypeptide. Instead, the glutamic acid is replaced with valine. Okay, so what happened to the sickle cell? What happened to the red blood cell? It has a sickle cell beta globin. Okay, now another example here is this one, but normally in exam they will ask for this particular example. Okay, sickle cell disease, which is caused by the missense mutation. You must know that. Okay, this is another example of missense mutation. Okay, where you have, okay, T, okay, instead of C. okay, if we refer to this diagram here, okay, it should be C C G. But what happens? The C is replaced with T, so it becomes T C G. Okay, and by right the mRNA will code glycine. What happened? Serine is being coded instead. Okay, so now the third example, okay, of point mutation is nonsense mutation, which is substitution, okay, that convert an amino acid specifying codon to a stop codon. So meaning to say, by right you should get an amino acid, but what happens? You get a stop codon instead, right? So this will cause translation to be terminated prematurely, which will result polypeptide to be shorter than the polypeptide encoded by the normal gene, and this will lead to non-functional protein, obviously, right? By right you should get a long stretch of polypeptide, okay, but instead you get a very short polypeptide, and a short polypeptide will lead to non-functional protein. So example is, okay, TTC on the DNA template strand. Okay, this TTC here mutated to ATC. ATC. What happens? The T is being replaced with A. Sorry, the T is being substituted with A. Okay, so what happened? The mRNA codon that used to be AAG, okay, that codes for lysine, okay, what happened? The mRNA becomes UAG, that codes the stop codon, and this nonsense mutation, okay, nonsense mutation is shown in beta thalassemia and cystic fibrosis. Okay, now that is about point mutation. Now let's continue with frameshift mutation. Okay, this involve nucleotide base pair being inserted or deleted. All this while we have discussed about substitution, penggantian. Okay, the nucleotide base pair is being substitute, replace, ganti. Now what happens when this nucleotide base pair is being inserted or deleted? Okay, so insertions and deletions are additions or losses of one or more nucleotide pairs in a gene. Okay, and this will alter the reading frame in the mRNA downstream of the mutation. All nucleotides downstream of the deletion or insertion will be changed or improperly grouped into codons. Okay, and the results will be in extensive missense and extensive nonsense. Okay, now class, downstream, upstream. Okay, now here the region of this DNA here is known as the upstream. Okay, and when you are headed that way, okay, this is the direction of the translation. This is known as the downstream. Okay, oh saya tulis, okay, downstream. So now let's say this is the point where the nucleotide base pair is being inserted or missing. Okay, any sequence of codons here downstream, okay, from any nucleotides downstream of this deletion or insertion, okay, is already changed. The sequence of the nucleotides here already changed because you have an addition or losses of a nucleotide base pair. And class, nucleotide pair insertions or deletions of prime shift mutation, it can involve one nucleotide base pair or more, okay, two base pairs, three, and so on. Now, extensive missense, okay, this occurs when there is a deletion of base pairs, okay, base pair or base pairs. Example is AAA on the DNA template strand. Okay, let me change the color of the pointer. Okay, yellow, orange. Okay, now example is the AAA. This AAA on the DNA template strand, it loses or missing one A here. Okay, what happened? The DNA will be mutated to AAC. Okay, AAC. The C from the previous, or the C from the downstream, it is shifted to the upstream. Okay, sorry, this C here, okay, shift to the upstream A. Okay, so the C nucleotide base comes from the adjacent downstream. Okay, betul kan? C ini, yang ini C nucleotide base pair, dia daripada adjacent A ini, and this C ialah downstream. So the mRNA codon that used to be UUU, you can see from here, right? Should be UUU, and GGC. If you follow the sequence, right, UUU, GGC, and UUAA would become UUG. You see, UUG and GCU. That codes the leucine and alanine instead of phenylalanine and glycine. Okay, can you see the sequence of the amino acids already changed? Okay, and most probably, okay, this polypeptide will lead to protein that is non-functional. Okay, so this is extensive missense. Okay, another one is immediate nonsense, okay, or premature termination. This is caused by insertion of at least one nucleotide base pair. Now, example is the TTC on the DNA template strand, which has an extra A being added here. Okay, so the extra A is added somewhere here. So the DNA template strand will be mutated to ATT, whereas it should be TTC. So ATT. So the C here, what happens, because initially it was TTC, right? So this C here, it will shift to the downstream, adjacent downstream. So the mRNA codon that used to be AAG, okay, and UUU would become UAA, which codes the stop codon to terminate the translation instead of coding lysine and phenylalanine. Okay, so what happened to the polypeptide? It is shorter. Okay, and a short polypeptide will lead to non-functional protein. So this frame shift mutation, it has disastrous effect on the resulting protein more often than the point mutation or nucleotide pair substitution. Now, another example here, okay, or case study here is a 3 nucleotide pair deletion and 3 nucleotide pair insertion. Okay, this case here, this 3 nucleotide pair insertion or deletion will not cause any frame shift mutation. Okay, but what happened? It will cause the losses of amino acid or the addition of an extra amino acid, and this further, okay, will lead to non-functional protein because the polypeptide is non-functional, because the polypeptide either it loses one amino acid or it gains an extra amino acid. So what will happen to the protein? It will be non-functional. Now an example here, okay, is the TTC on the DNA template strand. Okay, TTC, so it should be here, TTC. Okay, example is the TTC on the DNA template strand missing. Okay, this TTC when missing is shown here. Okay, it's being removed. Okay, so what happened? The AAA, which is downstream of the TTC, will be shifted upstream. Okay, it will be shifted to the TTC position. So this will lead to the missing of one amino acid in the polypeptide, causing the polypeptide to be non-functional. Okay, by right you should get lysine after methionine, then only phenylalanine. Instead you get phenylalanine after the methionine. Okay, missing of lysine. Okay, and this is shown in thalassemia major. Right, now there is no example, okay, for insertion of a 3 nucleotide pair or 3 nucleotide base pair. But I just want to stress out that an insertion of a 3 nucleotide pair or base pair will lead to an extra amino acid in the polypeptide, which cause the polypeptide to be non-functional. Okay, now test your knowledge. Okay, I believe you have these questions. Okay, so pause the video, try to answer this question on your own. Okay, then you come back here. All right, done? All right. Okay, why might a point mutation in the DNA make a difference in the level of protein's activity? Point mutation, why? Because it might substitute an amino acid, okay, in the active site. Substitution. Okay. Which of the following types of mutation resulting in an error in the mRNA just after AUG start of translation is likely to have the most serious effect on the polypeptide product? Is a deletion of 2 nucleotides. Okay. What is the effect of nonsense mutation in a gene? Okay. As you see, it introduced a premature stop codon in the mRNA. Remember, nonsense mutation. Okay, now these two questions here. This one I want to discuss with you. Okay. Now, if each of the following options, okay, is a modification of the sentence, the cat ate the rat. Which of the following is an analogous to a frame shift mutation? Okay, frame shift mutation. What is frame shift mutation? Insertion or deletion of at least one nucleotide base pair. So now the cat ate the rat, you have to search here between the options A until E, which has insertion, okay, or deletion of one or more nucleotide base pair. The rat ate the cat, this is translocation. The rat, okay, it was previously from downstream shifted to the upstream here. Okay, the ate, the same goes for B. Okay, and C, the cat are the rats. Okay, so it can be C. Okay, the R is being substitute. The cat ate the rat. Okay, now D, the cat ate ATE. Okay, one is missing here. Okay, one is missing here. Sorry, this is substitution, right? C is substitution, so this cannot be frame shift mutation. It is point mutation, right? Substitution. This is, however, D is in D. Okay, one nucleotide base pair here is missing. If you look at E, cat at the rat. Okay, cat at the rat. Cat at the rat. So the is missing. Okay, now the, it involves three nucleotide base pair. Remember earlier I mentioned three nucleotide base pair. It doesn't cause frame mutation. So D would be probably the answer. Okay, this is frame shift mutation. Okay, is it D? Alright, now what about this question here? You try to answer on your own first. Okay, I'll give you 3 minutes, think about it. Okay, done. Now, which of the following is analogous to a single substitution mutation, like what I've mentioned before this? Okay, so the answer will be C, right? Because R is being substituted here. Okay, sickle cell disease, probably the result of which kind of mutation? Sickle cell disease, is it point mutation or frame shift? Okay, it's point mutation. Frame shift mutation could result from which one? Frame shift mutation. Okay, deletion of 3 consecutive bases. Oh, cannot be. Frame shift, a deletion of 3 consecutive bases, this will not cause a frame shift mutation. Frame shift mutation will only be caused by one or two nucleotide base pair. Okay, incision or deletion. Okay, so the answer would be E. Okay, so yeah, need to rectify the answer here. So the answer here is not D but E, either an insertion or a deletion of a base or two base pair. Okay, remember, three base pair, insertion or deletion of three base pair, it doesn't cause frame mutation. Okay, why point mutation would be most likely to have a catastrophic effect on the functioning of a protein? Point mutation. Why? Okay, base pair deletion near the start of a gene. Yes, what happens if a base pair is deleted near the start of a gene? It will cause a premature polypeptide, a short polypeptide. A short polypeptide will lead to a non-functional protein. Okay, so that's it for today. Okay, happy studying. Bye. Thank you, Michael.